Hi. In the second part of Introduction to Nervous System, we will learn about the components which form the nervous system and also the structure of the excitable cells or the neuron. Objectives of this session are: at the end of watching this video, you shall be able to classify the cells which form the nervous system, describe the structure of a neuron, describe the shape or appearance, location, and function of each organelle of neuron, compare and contrast the features of dendrite with that of axon, describe various terminologies used with respect to aggregation of neurons or its parts. Let's get started with the cells of the human nervous system. The components or cells of the human nervous system can be broadly grouped as the excitable cells or neuronal cells, which are commonly referred to as neurons. The non-excitable or the non-neuronal cells are referred to as the neuroglial cells. The neurons are responsible for encoding the information, generating an action impulse, and also transmitting the information to other neurons or even non-neural cells. Whereas the neuroglial cells do not generate action potential but can convey information encoded as changes in calcium concentration within the cell. For decades, there has been a belief that the number of neuroglial cells exceed the number of neurons by ratio of 10:1. For every single neuron, it was believed that there were 10 neuroglial cells. But this has been proved wrong. Recent researchers suggest that the number of neuroglial cell is almost same as that of the neurons in human nervous system. Though in thalamus, the ratio of neuroglia to neuron is still about 17:1. Except thalamus, other parts of the human nervous system is believed to have the same number of neurons and neuroglial cells. The structure of the neuronal cells or excitable cells, commonly referred to as neurons. A neuron can be broadly divided into two parts. One, cell body, which is otherwise referred to as soma or perikaryon. Then we have the processes, which are referred to as neurites. Neurites can be dendrites or axons. Let's learn about the structure of a neuron in detail now. As we discussed earlier, a neuron has a cell body. The shape and size of the cell body greatly varies. The size of a neuron can vary anywhere between 5 microns to 100 microns. This is the cell body of a neuron. Let's try to learn what are the organelles which are present in the cell body. The cell body has a round and central nucleus with a prominent nucleolus. The prominent nucleolus suggests the increased activity of the neuron and also suggests that neurons are cells with high protein synthesis. Apart from the nucleus, we can make out many cytoplasmic organelles. These are, we can appreciate mitochondria, which are scattered throughout the cytoplasm of the neuron. So these are the mitochondria, and as we know, the function of mitochondria is to provide the chemical energy. We have the Golgi complex. Golgi complex appear as clusters of flattened cisternae. These are the Golgi apparatus which lie close to the nucleus. The function of Golgi apparatus is they act as the packaging organelles. They help in transporting the products to the nerve terminals. They also help in formation of cell membranes. The characteristic feature of the cytoplasm of a neuron are organelles referred to as Nissl substance. Now this Nissl substance are scattered throughout the cytoplasm of the neuron. Now these nissl substance are polyribosomes which are associated with rough endoplasmic reticulum. The function of these nissl substance is believed to be synthesis of protein and neurotransmitters. Even though they are present in cytoplasm, these nissl substance they extend into the dendrites, but they never extend into the axons. The next cell organelle is the lysosome. Now, lysosomes are membrane-bound vesicles which are again scattered throughout the cell body of a neuron. They usually measure about 8 nanometers in diameter and they function as the cell scavengers. They help in intracellular digestion. Centrioles. The centrioles appear as hollow tubular structures which are paired. Now these are confined again to the cytoplasm, do not extend into the processes. Remember, the peculiar function of centrioles here is not to assist cell division but to maintain microtubules, the cytoskeletal elements. Now these cytoskeletal elements include neurofibrils, microfilament and microtubules. The neurofibrils appear as linear fibrils which extend from the dendrite into the axon. There are number of parallelly arranged linear fibers and they extend from dendrite to axon. Now these neurofibrils help in determining the shape of the neuron, microtubules. The microtubules appear as linear tubes which run between the neurofilaments. These microtubules assist in cell transport. The third type of cytoskeletal element present is microfilaments. The microfilaments appear as linear fibers or linear fibrils and they are present beneath the plasma membrane of the cell body. Now these microfilaments assist in formation and retraction of cell processes and they also assist in cell transport. The cytoplasm of a neuron can contain pigments which could be lipofuscin or even melanin. Now these pigments could be the byproducts of metabolism or related to formation of neurotransmitters. The areas of nervous system where we often find neurons with these pigment granules are the substantia nigra in midbrain and also the locus coeruleus. This is regarding the structure of the cell body or the perikaryon and each of its organelle with its location, shape and function. Now, let's learn about the neurites. Dendrites are short processes which arise from the cell body and taper towards the end. Close to the end, they show extensive branching. These are the dendrites. The important thing about these dendrites is the external surface of these dendrites have projections coming from their external surface and this projection is referred to as dendritic spines. The function of these dendritic spines or even dendrites is to increase the surface area. As we discussed earlier, the cytoplasm of dendrites resembles the structure of cell body and contain almost all the cell organelles including the nissl granules. Now let's learn about the structure of the axon. Axon is the longest process which arises from the cell body. This arises from a conical elevation, the axon hillock. Axon hillock is the conical area from which we have the origin of the axon. The axon hillock leads to an area referred to as the initial segment. Initial segment measures about 50 to 100 microns and this is the most excitable part of an axon, and hence this is the site where action potential originates. The plasma membrane which surrounds the axon is referred to as axolemma, and the cytoplasm of the axon is referred to as axoplasm. The axoplasm do not contain nacelle granules and Golgi substance. The terminal portion of the axon branch profusely, and each branch shows enlargement. These are referred to as terminal buttons or synaptic buttons. The axon will be covered by myelin sheath, which is formed by the oligodendrocytes in case of the central nervous system, or Schwann cells in case of the peripheral nervous system. So these are the myelin sheath which surrounds the axon. Remember, myelin sheath is absent where we have the axon hillock and the initial segment. The area between the two nodes, this is referred to as the node of Ranvier. Nodes of Ranvier are specialized regions where myelin is absent and hence action potential are generated. This is also the place where the axons can branch. If we take a section of this region and enlarge, this is the axon, then we have the myelin sheath. The territory of myelinated axon between adjacent nodes is called an internode. The region close to the node where myelin sheath terminates, this is referred to as paranode. The area just adjacent to the paranode, this is referred to as juxtaparanode. Importance of this area are the node of Ranvier has highest number of sodium channels, and hence this is a place where action potential is generated. The juxtaparanode has richest number of fast potassium channels. This is regarding the structure of a neuron. Let's learn a little bit about the process of chromatolysis. Chromatolysis is a phenomenon in which the nacelle substance or granules appear to have disappeared in response to neuronal injury or neuronal fatigue. But the fact is, the nacelle substance do not disappear, but instead they move towards the periphery of the cytoplasm of the cell body of neuron. This gives a false appearance of the disappeared nacelle substance. Let's try to compare the features of a dendrite and an axon. Dendrites, as we learned, essentially are the processes which carry the impulse towards the cell body. Dendrites are present in multiple numbers, they are not single. The shape of the dendrite appears to taper towards the end where the dendrite branches. The external surface of a dendrite is not smooth, but instead has projections which are referred to as dendritic spines. They contain majority of the cytoplasmic organelles present in cell body, including nacelle substance. Whereas an axon, as we learned, is a process which carry the impulse away from the cell body. Usually, axon is a single and the longest process of a neuron. The shape of the axon is tubular and uniform throughout, except at the terminal portion where it branches into synaptic buttons, where it terminates. The external surface of axon is smooth. Nacelle granules are characteristically absent in axon. Now, these are the features which will help us differentiate between a dendrite and an axon. Coming to the end, let's try to understand the terminologies which are used with respect to either parts of the neuron or neuron itself. Let's draw the outline of the cerebral hemisphere. This is the outline of the superolateral surface of the cerebral hemisphere. Then let's imagine this is the midbrain, pons. Then we have the medulla, and then we have the continuation of the medulla as the spinal cord. When the cell bodies of neurons form a collection which is wide and dispersed, we call this aggregation as gray matter. So, aggregation of cell bodies of neurons within the central nervous system can be referred to as gray matter when it is widely distributed. At the same time, when the collection is localized, it is referred to as a nucleus. Now, the nucleus can be in the brainstem or it can be in the spinal cord. Then, the third aggregation, which occur in the brainstem, where the cell bodies appear as collection in the form of elongation. These are referred to as columns or layers. So, in central nervous system, collection of cell bodies can be referred to as gray matter, nucleus, and then column. When collection of cell bodies occur outside the CNS, for example, we have the spinal nerve coming from the spinal cord, and the spinal nerve has an enlargement referred to as ganglion. Right? When we take this ganglion, even this is made up of aggregation of cell bodies of neurons, but now this is referred to as a ganglion. So ganglion refers to collection of cell bodies of neurons outside the central nervous system. In central nervous system, if we have aggregation of the neurites or the processes, it is commonly referred to as the white matter, or it is also referred to as the nerve tract, which is commonly seen in the spinal cord. Now, if this aggregation occurs outside the CNS within the peripheral nervous system, we refer it to as peripheral nerves. Knowledge of these terminologies is very essential to understand further concepts of neuroanatomy. To summarize, the cells forming the human nervous system can be divided into neurons and neuroglial cells. Neuron has a cell body and neurites. Neurites can be dendrite and axon. Cell body has all the organelles which are typical of a human cell. Along with that, nissl substance is a characteristic organelle present in the neuron, responsible for protein synthesis and neurotransmitter synthesis. Apart from nissl granules, the cell body has mitochondria, Golgi apparatus, lysosomes, microtubules, neurofilaments, microfilaments, pigment granules. We also discussed about the contrasting features of dendrite and axon, and then we discussed various terminologies like gray matter, nuclei, column or layers, white matter, nerve tract, and peripheral nerve. Thanks for watching. If you like the video, leave a like. Please leave your doubts in the comment section. Share and subscribe. Thank you.